Hello, my name is Julia Guerro. Here is my name and the date, and I'll be presenting today on how the pancreas and liver affect digestion. Hello, my name is Julia Guerro, as stated before, and I will be presenting on how the pancreas and liver play a role in digestion. The pancreas contains exocrine and endocrine secretory epithelium and is responsible for the secretion of multiple different enzymes that aid in digestion. At first, many of the pancreatic enzymes are secreted as something called a zymogen, which are secreted inactive. They get activated after trypsin, which, after activation by enteropeptidase, trypsin starts as trypsinogen and then is activated into trypsin. Trypsin activates the zymogens, and that's when they begin to do their job. There are multiple different signals that start the release of enzymes from the pancreas, and some of these signals include distension of the stomach, undigested food, neural signals, and the gastrointestinal hormone CCK. They all stimulate pancreatic enzyme secretion. One of the important secretions from the pancreas is bicarbonate, which gets secreted in the duodenum, and it neutralizes the acid that enters into the duodenum from the stomach. Starting with the exocrine secretory epithelium in the pancreas, we have acinar cells. And the acini are lobules in the pancreas that are actually very similar to the salivary glands that are in the mouth. The ducts that come from the acini empty into the duodenum, and that is where the sodium bicarbonate, as stated in the last slide, are secreted from. And the acinar cells themselves secrete digestive enzymes that assist in digestion. A few of these digestive enzymes include lipase, protease, and amylase. Now for the endocrine secretory epithelium, we have these types of cells called islets, and they're responsible for the secretion of insulin and glucagon. Insulin is secreted after a meal, and the purpose is to decrease the blood glucose, so it stimulates the liver to store the glucose into glycogen and put it in storage. And this takes the glucose out of the bloodstream, or the excess glucose, I should say. Glucagon is responsible for the opposite, so when you haven't eaten a meal in a while, glucagon stimulates the liver to break down the glycogen that has been stored into glucose, and it increases the blood glucose levels. The liver also has a very important role in digestion, similar to the pancreas, where it's responsible for secretion of various different things. Starting with the liver cells themselves, they are called hepatocytes. They are responsible for bile secretion. And bile secretion is very complex. There's a lot of different parts. I have a few of them listed here. But bile salts, bile pigments, and cholesterol are the main parts of bile secretion. Bile is a non-enzymatic solution that consists of the three things I just listed, and it aids in the digestion of food and proteins and things of that nature. Starting with bile salts, they facilitate enzymatic fat digestion, and they act like a detergent that make the fats soluble, because obviously a lot of our membranes are lipid insoluble. They are made from steroid bile acids combined with various amino acids. Then going into bile pigments, they are waste products of hemoglobin degradation that occurs during digestion. And cholesterol is another waste product that is excreted in feces. The liver works very closely with the gallbladder, and when the bile is secreted by the hepatocytes, it travels to the gallbladder via hepatic ducts, where it is stored and concentrated, and once that process is done, it is then secreted into the intestinal tract with the rest of the enzymes, and that's where the final digestion and absorption takes place. The study exercise I've created is to create a concept map of all the important topics covered in this presentation, and whether this is sort of like a treasure map or like a brain dump type thing, something along the lines of showing the different steps and what's connected to what. And I think that is very helpful in studying. And I also included that at least 2 drawings must be incorporated into the concept map, because when I was studying for this presentation, I did a lot of drawings of the actual pancreas and liver and the steps associated with their contribution in digestion, and I thought it was very helpful for my own understanding, and so that's why I included it as a part of this study exercise. I think the concept map and the drawings will help further a deeper understanding of how these 2 organs play a very important role in digestion. And so some of the important things that I want to be included in the concept map are the hepatocytes, islets, acini, insulin, glucagon, bile secretion, zymogens, and trypsin. And I want all of them to be displayed and shown how they are connected or not connected, and their relationship to each other and to digestion. And I think that this will aid a deeper understanding of the topic. Here is my work cited, and I pretty much just used the textbook for this presentation, so I cited the textbook. And this presentation is now over. Thank you.